Welcome to this module of the Big Data Analytics Summer School for the Random Forest track. So in this module we will discuss different properties and abilities of random forests. So first we'll talk about variable importance, different kinds of measure, how to measure variable importance in models. Then we will talk about sample proximity, which can be used for clustering or model assessment or quality control. Then we will talk about the ability to actually mix variable types in random forest models. And finally, we will shortly talk about the ability to parallelize training and testing. So one of the intrinsic properties of random forest is the ability to measure how much various features actually contribute to the classification task. So how different features improve the prediction accuracy of these models. So in practice that's done by, after learning a random forest, one can use the out-of-bag data to estimate the out-of-bag error as described in one of the previous modules. And now we can go ahead and actually permute features one by one and then measure how the performance of the random forest ensemble goes down. And the more it goes down, the more important the feature was. So in this example we permute feature I. And this feature, for example, was used 3 times in the forest at split, or in this tree of the forest, at split nodes 1, 6, and 9. So by permuting during testing, one would take just a random direction at each of these nodes. And naturally the outcome would be much worse, or at least worse to some degree, based on the importance of that feature. On the right side, you now have a measure for this variable importance. So here it's a ranking of features for spam detection from one of the standard spam databases. So this is important because it gives us insight into the model. Depending on the scientific scenario, one is more interested in actually how the model works, or one is only interested in prediction accuracy. For example, in most computer vision scenarios, we do not care which kind of pixels or rectangles or textures are used. We are only interested how well can the robot, for example, navigate some kind of terrain based on the visual prediction. On the other hand, if we have cancer research scenarios or biological scenarios, we are more interested which genes or which proteins actually contributed to classifying a patient for having cancer or not. So for these tasks, this variable importance measure, which comes very cheaply computationally, is an enormously important ability of random forests. The next topic we talk about is something quite interesting because that's something most other models can't do, and that's sample proximity. So when we actually test, again, samples we didn't use for training, these could be these out-of-bag samples or samples from a different test set, we could actually check in which leaves of the tree they end up. So in that scenario, we have 3 new samples, 1, 2, and 3, and we drop it down this single tree, and 1 and 2 end up in the same leaf, whereas 3 ends up in a different leaf. So we now can actually collect this information in a proximity matrix. You see here in the rows and columns are the indices of these samples, and in the cells we have counts how often the samples end up in the same leaf. So just 1 for the samples 1 and 2, and 3 does not share a leaf with one of the other samples. So for one tree that's not very exciting, but imagine you have actually an ensemble of trees, so a whole forest, and you do that with every tree in the forest, you come up with a matrix which nicely depicts how similar samples are based on the view of the random forest. So here in that case we would have 83 times 1 and 2 ended up in the same leaf. So in 83 trees these two samples ended up in the same leaf. 3 times sample 3 ended up with 1, and 11 times with number 2. So we could conclude in that scenario that 1 and 2 are quite similar to each other, but 3 is not. The interesting property here is that that gives us actually a clustering of the samples, again in the view of the classifier. That's especially an interesting property if you are in very high dimensional feature spaces. Let's say you have a few hundred patients, but you have hundreds of thousands of genes, and not all of these genes are of importance. So you actually want to have, first of all, a subselection of these genes, which the random forest does because it is invariant to noise features, and then based on these genes which are important to the classification, for example between cancer and non-cancer, you can get this view of the proximity. So here's a practical example from the cancer research task in a module farther ahead. So here we classified 500 cell nuclei. So on the left side you have these raw proximities of 500 cell nuclei. So the indices for the nuclei on the x and y axis, and that gives you a similarity matrix. So that's a standard mathematical object. It gives you the distance between these objects or the similarity between objects. We now can cluster that matrix with some kind of clustering algorithm. It could be something very simple like standard walled clustering or hierarchical clustering, agglomerative clustering, or something more advanced like a Vischer-Dirichlet clustering, which does a nice partitioning in this case. And you can see the result on the right side. So what you can see here is that actually the cell nuclei nicely split up into 3 classes, 1, 2, and 3, which are nicely different. And then we have kind of muddy classes 4 and 5, where 4 is mixed most of the time with 2, and 5 is mixed with 4, 3, and 2. So in this case, it's actually a binary classification case. So it was cancerous nuclei versus non-cancerous nuclei, but we find these 5 different subsets. We now can take actually a look at these samples. So what you see here is for every cluster you have a row, and you see a number of cell nuclei randomly sampled from these 500 cell nuclei. And on the right side, you see the class distribution between malignant and benign, cancerous and non-cancerous. And interestingly, in the view of the classifier, you have clusters like cluster number 3, for example. Cluster number 3, nicely separated from the other cluster, and it really contains only cell nuclei which look very similar. So you have this large, roundish cell nuclei which are malignant and which are a clear indication of renal cancer in that case. And they visually look different. So these proximities in the random forest give us a clustering which really ends up with visually differently looking nuclei. For example, 4 and 5, these clusters are very mixed, so you have completely different classes in these clusters, malignant and benign, and as you can see, these patches look quite chaotic. And it's even for experts quite often hard to decide what these nuclei are. So as a conclusion, that step could actually be used for quality control. So imagine a setup where you get new samples, process it in that way, and then you could reduce the analysis to the clusters which only produce very clear malignant or benign samples, or at least very consistent looking samples of nuclei. The result would be probably a better outcome than processing a whole histological slide where you have a lot of blurred areas as seen in cluster 5, or overstained areas as seen in cluster 4. So in practice, proximity and clustering based on random forest is something which actually no other method can do out of their standard application. So that's a very useful and nice property of random forests. Another ability which is very useful in practice is the ability to mix data types in random forests. So normally, or in the modules before, we only talked about continuous variables. Let's say the height of a person or the expression of a gene. But in practice, we have a lot of different subtypes of variables. We could have categorical variables, like countries or cities, or we could have ordinal variables where we have an ordering on something. We could also have more complicated structures, so some mathematical objects like matrices or vectors, which are the samples in that case. And what you can do in a random forest is you could use different kinds of features at different split nodes. For example, at the beginning at node 1 and 2 we have continuous features, where the feature is just checked against a threshold, which is learned during training. For categorical features in node 3 and 5 in this example, we would check, for example, if a feature is equal to a category. For example, here feature 13 is equal to USA, so feature 13 could be the country. Or, for example, if it's part of a set. So in that case, if feature 66 is part of the set of LA, New York City, or Graz. And then again, for ordinal features, they behave quite similar to continuous one, but you only would have ordered categories, for example, integers in that case. So and you can mix all these features in a single model without pre-processing. If you would use a support vector machine or out-of-the-box lasso approach, you would have to build contrast variables based on these categorical variables and somehow overcome the limitations of these mixed models. That's an ability which is especially important for business applications. Imagine you have these huge sales databases where you quite often have very mixed variables, like country codes and so on. So finally, I want to talk about parallelization. So there are different ways how to parallelize random forest. There's a very straightforward way, which would be parallelized based on trees. So you could learn trees on different processes in one machine, or if you have an MPI set up or something similar, you could train different trees on different machines in a cluster or in the cloud, for example. Another possibility is actually to try to evaluate the features in different processors. So for every split, we would evaluate different thresholds, for example, for continuous features. Also, that step could be parallelized. Or in computer vision, it could also be simply parallelized over areas of an image. In addition, this ability makes them also quite straightforward to implement for GPUs and FPGAs. So in practice, there are highly optimized variants of random forests for these kinds of hardware platforms. And one example is Microsoft's Kinect, where they have an implementation for skeleton recognition. Or in our case, on the bottom is an example from JPL. Here we are working on an FPGA implementation of random forests, and that will be used at one point on Mars or different missions to characterize different kinds of terrain. So the goal here is to really have a camera together with the random forest algorithm on an FPGA, so all the processing is done at once, and this science instrument actually gives as output a texture classification of a scene, which then can be used for autonomous driving or for detecting scientifically interesting targets. So all in all, these properties make random forests very good off-the-shelf method, which has a lot of application. In practice, naturally, one would have to optimize for one's own application, also choose different algorithms, but in this case, at least at JPL and Caltech, we have a whole set of applications where they proved very suitable. Thank you.